Welcome to Own Your Heart Health. I'm Dr. Regina Druz, your holistic cardiologist. This week, we will dive into common heart health concerns, uncovering root causes, and unpacking scientific discoveries and controversies. The information provided does not constitute medical advice. Please contact your healthcare practitioner before making any changes that may impact your health. Hello, listeners. I am so impressed that you tuned in to yet another episode. And this episode is an amazing treat for me and for you. I'm here with Dr. David Furman. And Dr. David Furman is actually joining us from the West Coast. He is a societal entrepreneur as well as a very prolific researcher whose goal is to redefine how we approach biomarkers of longevity, right? And how we can better ourselves by really looking at testing, which allows us to choose the best actionable steps for ourselves so that not only can we enjoy an extended lifespan, but hopefully an extended health span. Dr. David, welcome to the show.
Thank you so much. And thank you for having me, Regina.
You're welcome. And I am going to ask you a question that all of my guests and my listeners know as well. I always ask everyone, how did you grow up to be who you are today? Kind of tell us your story.
Okay, so I was born in Argentina in South America, and I moved to Israel when I was very young. And I spent a few years there. My career started really when I was probably like 17, 18. I was not really sure what to do with my life. I was in arts and mostly music. So I'm a musician, and very like the creative side of my brain, right, was exploding. And I talked to my dad, and because, I wanted to create impact. I wanted to create impact in humanity and do better. I think that we human beings that are intelligent, we have a responsibility to really try and change the way the world is at the moment. We know, there's a lot of issues in the world, and try to create impact. And so I told him I want to study medicine, and help people do better with their lives and with their health. And he convinced me to study biology, because if you do biology or biochemistry, you can really have a much stronger impact in humanity than if you're maybe, fixing one by one as a doctor is great, a lot of satisfaction. But if you can fix problems of many, maybe hundreds of thousands or millions, is much more impactful. So I decided to do biology, then I went for immunology, then I turned into data scientist. I moved to Stanford. Stanford recruited me while I was doing finishing my PhD. I did my postdoc there. And after looking at a lot of data from 1,000 Immunomes project that we founded in 2007, I realized that a lot of the signal came from the aging, the biology of aging. And that's when I turned into health and longevity and aging research around in 2012. So I'd say, over 10 years ago, over 15 years ago-ish.
Wow. So this is very impressive. You put in a lot of words, and, as my listeners know, is that I always want to break things down and unpack them so that people understand. So, there are a couple of really important points, right? People may or may not know is that one of the primary systems that ages when we age is our immune system, right? So you mentioned 1,000 immunome projects. So what's happening with the immune system, number one, and number two, what is the 1,000 Immunome Project, and what did it actually, why was it launched? What did it find? Who did it study? All of those things, if you could expand on that.
Right. So one of the most important systems in the human body that determines rates of aging in other systems is the immune system, because we're sensing the environment and we're reacting to it. And that causes what we call systemic chronic inflammation. right?
Mm-hmm.
And so inflammation is the enemy, silent enemy that is pushing cardiovascular disease, Alzheimer's disease, musculoskeletal conditions, metabolic disorders, all of them, right? In the year 2000, we didn't know too much which biomarkers were associated with systemic inflammation and which ones were causal. What we're seeing now, after maybe 25 years of research, is that the more we learn about the immune system, and the more we map these biomarkers of immune system functioning and inflammation to diseases, the more we can realize that it's really a hub for
Mm-hmm.
aging. So hallmarks of aging, as many people
Mm-hmm.
know, the cellular senescence and epigenetic changes and all of those are largely driven by inflammation. And so that's
Mm-hmm.
how this 1,000 Immunomes program started, to try and redefine chronic inflammation and give some, solid science behind. We recruited 1,000 people. We secured over 70 million from federal agencies. And this study ran for about 15 to 17 years. And so we're discovering biomarkers of systemic chronic inflammation that predict morbidity and mortality. So that's how the 1,000 Immunomes project began.
Interesting. For our listeners, so those people were volunteers. Were they patients? Were they coming through, Stanford
Volunteers,
healthcare
yeah,
system?
for,
Volunteers.
for completely relatively healthy individuals, ambulatory, that came from a flu vaccine.
Flu vaccine. So they, if they had any disease, let's say hypertension, heart disease, were they excluded or were they included?
They were excluded unless they had controlled some of the diseases that you cannot avoid. And many people over 65, you get to see.
Right.
Metabolic syndrome, for example, is a very typical one. But if it's under control, they were included. And many diseases happen during the study, right? So we could
Right.
prospectively predict who could, kind of develop different conditions as the study was developing.
Oh, perfect. So this is, to me, this is fascinating, right? Because we are so used to laboratory markers being blood markers, and some of them are very common, and they are often checked, like high sensitivity C-reactive protein and IL-6. And I think majority of adults, certainly most of our patients, they do know these numbers, right? They had these numbers checked. So why do you, the immune system aging and this chronic inflammation is not the same as what we are checking in the bloodstream? Can you
Yeah, so
explain the differences?
let's talk about acute versus chronic, because when we look at biomarkers of aging and we focus on inflammation, the first thing that pops up is, as you mentioned, hsCRP and IL-6 and TNF alpha. For the past, I would say, 2 decades, more and more research has demonstrated that those biomarkers are not necessarily associated with aging. They're very poor predictors of non-communicable diseases.
Mm-hmm.
Cardiovascular disease can be predicted a little bit better than flipping a coin with hsCRP. Most cardiologists now are not using it at all, but it's mostly being used in the infectious disease arena because it does change dramatically with an infection. So we have, on the one hand, acute infection or inflammation that is the response to, say, cut your finger, becomes red, swollen, infected, and you have high levels of hsCRP, IL-6. But when you look at in the blood, systemic biomarkers of chronic inflammation, you typically don't see IL-6, you don't see CRP, you see others. And because there was a lack of information, we decided to take an unbiased approach and study hundreds of thousands of parameters from these 1,000 individuals and identify the most predictive ones.
Interesting. So what did you find in those 1,000 individuals that, what are these biomarkers. And by the way, I had the immune system age with Dr. Furman's test, and it's super informative, but this is sort of like requires you to open up your mind and understand things a little bit differently, is that these biomarkers are pointing toward chronic inflammation, the whole inflammation process, which in turn, of course, affects other areas of the body, very commonly causing two things that we are almost always worried about: genomic instability, which means our genes are changing. And, if our genes are changing, we could be more prone to diseases, things like cancer, for example, and what's known as cellular senescence, basically attrition or death of the cells in our bodies. And, there are all these hallmarks of aging. There's 12 of them. I'm writing a longevity code book, so they will definitely mention they are front and center, but this is sort of a very abbreviated version. So, Dr. David, what did you find? What are those markers, and how do they actually tell us, sort of the gauge? How do they gauge where we are in terms of the chronic inflammation happening in our body?
Yeah, out of the other, probably, again, hundreds of thousands of biomarkers that we looked into the blood, right? Gene expression, proteins, cell frequencies, things that you can capture in the blood, metabolites. The most predictive of chronological aging and diseases when you run your predictive models are proteins. And so we decided to look into the protein data. So that's the first piece, right? For filtering, we take all the genes, take all the cells and the metabolites, and then use the proteins. Out of all the proteins, the most predictive was not IL-6. That's way far in the list.
Too far down the line.
Yes, no, not at all included. And then it was CXCL9, also called MIG. This protein circulates in the blood. It increases with age. As a chronic biomarker, it causes derangements in your cardiovascular system. So it really pushes your endothelium, which is the linen of your vessels, and undergoes senescence, cell senescence. And you have more ventricular hypertrophy of the left ventricle of the heart. And you have arterial stiffening in people with high levels of CXCL9. The other protein that came as being very predictive is called eotaxin, or CCL11. Eotaxin 1 is produced by the immune system in response to mostly what we breathe, the quality of our air. Wow. Air pollution, when you're maybe cooking and the vapors that come out, all of that, allergies, all of that affect your lungs, and you start producing in your the macrophages in the lungs, they start producing this protein, eotaxin. Eotaxin goes in the periphery, and it causes derangements at the level of the brain. So it really passes the blood-brain barrier very easily. It's a very small, what's called chemokine. And this chemokine or cytokine goes to the brain, and it does cause dysfunctional issues and associates with dementia and loss of memory and stuff like that. Then you have another 3 proteins, gamma interferon, which participates in immune system activation. You need to have that. And then you have GRO alpha, the same story. And the 5th one, which is super interesting, is called TRAIL. TRAIL sensitizes cells to undergo apoptosis. And these are senescent cells. So if you have low levels of TRAIL, you don't have enough apoptosis in your senescent cells, and they accumulate in the body. So overall, we're picking up different aspects of the immune system that contribute to different aspects of aging, right? Cell senescence, brain and memory and health in the brain, and cardiovascular health.
Cardiovascular. So that's cardiovascular, that's CXCL9 is produced in cardiac myocytes, or is it produced in macrophages that are infiltrating, let's say, coronary arteries or other arteries? Like, what is making it?
Yeah, typically it's produced by immune cells. As we go in the textbook and we see where the CXCL9 is produced, it's produced by innate immune system. right? And then it calls lymphocytes the site of infection. That's a textbook thing, right? But if you look at aging, these are produced by the endothelium. So the
endothelium itself.
And damaged endothelium produces CXCL9, which in turn damages more that the neighboring cells, and you start seeing this propagation of the senescence signaling, and then you have more and more senescence, and the tissue becomes stiff, more stiff, and then you have accumulation of high pressure for your heart, and you have hypertrophy, which is very difficult to restore.
So this is fascinating because we can measure endothelial dysfunction, right? We can certainly measure plaque progression. We can measure degree of ischemia. Recently, a lot of AI tools, we can actually discern plaque composition or even inflammation in the fat, let's say around the coronary arteries or around the heart chambers. There's a very strong linkage between that local inflammation and often it's called epicardial adipose tissue, which is basically fat around the heart. It's called EAT, or sometimes people call it ectopic adipose tissue, same acronym. So we can actually measure it. And this is what we're seeing right now, that these tissues, this EAT tissue, when it's surrounding the heart itself, actually is capable of infiltrating the cardiac muscle itself. So, and that results to stiffening of the heart and, a very specific syndrome of heart failure with preserved ejection fraction, which of course is linked to obesity and so on and so forth. So this is fascinating. So how early would you say, let's say, a person who wants to be preventative and they want to understand it, kind of what their trajectory is with cardiovascular disease and a very strong heart-brain health connection. How, at what age would you say they should start looking at these proteomic markers, right? Because what you're describing is essentially proteomics. And we could have an individual that has beautiful lipid profile and normal HS-CRP and no hypertrophy by ultrasound of the heart, but they probably already are showing these proteomic signatures of endothelial dysfunction and arterial stiffening. Is that correct? Or, what are you finding?
That happens all the time. It happens all the time. I've seen so many cases. We're working with different longevity clinics and concierge medicine doctors, and they're bringing their favorite patients, right? And they say, this is an incredibly healthy patient. Well, maybe not so much, right? There are inflammatory biomarkers that they didn't pick up using the regular, panels that you have available in the national labs. And you even have a whole study that we did in 150 people that were matched for the CRP, matched for BMI, matched for every single risk factor in cardiovascular health. And those with elevated inflammatory age, which is our metric, had arterial stiffening when you measure pulse wave velocity testing, central stiffening. They have increased left ventricular hypertrophy. And we're talking about normally healthy people that never hit the clinic for any issue.
Any reason, right?
Any issue with their health. They were selected to be the healthy population, and yet they had this underlying silent condition that we picked up with the test. I would say in the early 30s, it's a good time for us to start looking at, because, the molecular and cellular changes that precede disease, right? They start to appear between 15 and 20 years prior to any disease, right? And so before you have clinical symptoms, there's an underlying molecular change. We call it molecular interception of disease that we can capture at the early 30s. And so that's kind of the age where I should start, everybody should start measuring and monitoring.
I agree. And I think probably even in people, at least in my practice, if there is a specific constellation of risk factors. And, occasionally we get patients who genetically, for example, the polygenic risk scores are high, or they have some very specific lipid or cardiometabolic aberration. And so despite the fact that they're healthy, we typically wouldn't really find footprints of end-stage disease, right? We may not even find soft plaques because there just hadn't been enough time for these to form. But these, the proteome is fascinating because it is an early marker, and it could potentially forecast development of these diseases 10 to 20 years ahead, which is fascinating. And, I always tell my patients and anyone who is willing to listen that longevity is vascular. And we have proofs of this every single day, probably the major proof being that cardiovascular continues to be the dominant reason for mortality pretty much in the US and around the world. So the question always comes up and is, okay, so let's say we measured it. Is it something that could be changed? And what are you finding, based on your experience? And I know you developed IH and other tests, what are the most powerful interventions? What are we able to change? What is the timeline of these changes that a person potentially may encounter? Kind of, let's make it a little bit more clinically applicable.
Yeah. There's several layers, right? The first layer that we call one size fits all. That is your baseline. The baseline that everybody should fix from day one. Otherwise you don't get any benefit from layer two, right?
Right.
And that layer one includes things that we all know. Get good sleep hygiene, move around. Not sedentary raises inflammation. We know that. We've measured that. Go to nature. If you're sitting in a chair inside your office all day, that increases inflammation. We know that being sitting in a chair is a predictor of mortality long term, right? So we're trying to move
worse as bad as smoking, right? So
exactly. And so looking at the green in nature, being in a bonfire, all those things are papers, really strong papers looking at inflammation and how that decreases. Now let's maybe think about how we evolved, right? We evolved in a relatively low inflammation environment. We had spikes of inflammation when you get an infection, but you don't have the inflammaging that you see in our industrialized societies. So, wheat, anything from bread and pasta, that's inflammatory. Dairy products are inflammatory. We try to always keep a healthy diet with whole foods that are rich in nutrients. A great thing is all the berries. Berries are terrific. Fish is terrific. Small fish.
As long as it's mercury free, right?
small fish. You don't want those that accumulate too much mercury. So there's a lot of, and then in plastics, don't store in plastics because that goes into your brain and the heart, and they accumulate microplastics and nanoplastics. Now we know that they circulate in the blood and they accumulate in the tissues. So all of these things that we already know and you're probably very aware of and you're telling all your patients to do, right? The second layer is more the fine tuning. If I know that a specific protein is deviated from normal, I also want to know which environmental factors are triggering this protein. I alluded before to eotaxin and how that increases in response to air pollution. Right. So that's an excellent example of if you have deviations in eotaxin, you should monitor the quality of the air in your household, in your workplace, around your area, right? And try to have air purifiers, right? That's just one example. And there's many like that. So there's a fine tuning for each one of the proteins that are deviated. And then there's also like the more real world evidence, if you want, right? So we have now two new technologies, one that can measure aging of your organs, based on regular lab biochemistries. So you can go to your favorite vendor, Superpower, Function, all of those, and get your labs, and we can run the analysis, and that predicts the organ aging of each one of your organ systems. And so we've seen in those papers and in that research that acceleration of aging in the most organs is correlated with the quality of life you have, the amount of exercise. For example, if you eat white bread, that's like the top contributor to
Darn
accelerate.
it! I was just about thinking about my French baguette, but you're like taking it off the list. Okay.
Well, I mean, I think that the chronicity of the exposure is the risk factor. The risk factor is not what you do once a month, once in a while. You go, you have your pizza with your beer. That you do it, I don't know, once a month. That's fine, right? Because inflammation, if you have the buffer, if you do in a regular basis what the baseline that we were discussing, you have buffer. You can go back and
you have resilience.
Exactly.
I always tell my patients that there is resilience because a lot of the times, we're humans, we want simple solutions, right? And a lot of the times myself and even my patients, oh, there is this supplement or there is this technique or there is this peptide. But the bottom line is that the foundation has to come first. If the foundation isn't there, you are not likely to experience the benefit, potentially could even experience a negative effect. And it's almost like, what's the word, like peeling the layers of the onion, right? Until you finally get to that core. And that's what actually tells us how intense or how is our intensity of lifestyle effort matched to our level of exposure, and is there anything else that we're supposed to do? And I think it's fascinating because patients often come in and they say, I do everything right. I exercise, I eat right, I don't smoke, I sleep well, I don't stress as much, I really do all of those things. Why is my cholesterol high? Why do my joints hurt? Why am I still hypertensive? I'm so disappointed. I cannot possibly do more than what I'm already doing. And this is exactly the answer, is that, the sort of veracity of effort does not always match the cellular processes that are
100%.
happening in a person's body, right? We all know who was this famous French woman. I think she's still alive. She's like 119 years old, and she drank and she smokes, and and there are all sorts of things that are going on. So David, this is fascinating. Talk to me a little bit more about this organ aging. Is this an AI algorithm that takes regular blood work and sort of correlates it to these proteomic signatures you found in that immunome project? Like, how does it work exactly?
Right. Not exactly, but almost. So we use regular blood biomarkers, right? This is a comprehensive panel. These are 33 total biomarkers from cardiovascular and metabolic and all of those. We are agnostic as to where the biomarker comes from and the textbook mapping. Okay. We just take the biomarkers, all of the stuff we have access to in 500,000 people, and we're trying to predict mortality. So this is a mortality risk estimator that is taking mortality not as an all cause, but a system specific mortality. For example, if individuals died from cardiovascular disease or your Alzheimer's or dementia, some other mental conditions, or you have your musculoskeletal or metabolic, those are part of the International Classification of Diseases ICD-10 chapters. Right. And so we're predicting mortality in these ICD-10 chapters, and then we're transforming that mortality risk estimate in a biological aging for those systems. The assumption is that the older your system it is, the higher the risk to develop diseases in those organs.
But how do you adjust for the interactions, right? Because, vascular system, of course, interactions are multiple. Musculoskeletal system, is influenced by vascular system and so on and so forth.
Yeah, so I wanted to get to that. We're not supervising the interactions whatsoever. We're agnostic to the we're taking biomarkers that come from different tissues and organs, and we're trying to maximize the accuracy in predicting this mortality, right? So, for example, neural conditions, mental conditions can be influenced by inflammation. They can be influenced by your kidney function. So, for example, your creatinine is predictive. Your cystatin C, that is telling you about your kidney damage, it contributes to metabolic syndrome in our hands. And that's not what doctors are trained to capture. So we're using these algorithms. It's an AI-based algorithm that we train in 500,000 people with validated in 10,000 from the health and retirement cohort. And we're predicting mortality due to these ICD-10 chapters. And then we're transforming that to biological aging of each of the organs. And that can tell you what to do, right? It can tell you as a doctor, if this person has an Achilles heel and we're predicting that they may die from kidney, conditions, what to do already. Right. So
exactly. That's fascinating. And, how do you feel about, for many years, we've been fascinated between the interaction of environment and genetics, right? And the whole field of epigenetics is extraordinary. And so we've been taught about DNA methylation as one of the epigenetic mechanisms. And then it was sort of parlayed into the use of various biological age clocks and, first and second and third generation, and even the pace of aging. So is there a correlation? Because I personally, having done a lot of these bioage epigenetic clocks, I'm not very impressed because I have patients whose bioage by the epigenetics is stellar, but they're loaded with coronary artery disease. They had bypass surgeries. They have plaques in their carotid arteries. And, of course, the blood work in those situations very often is pristine. So you can't really put your fingers on it. So what is going on? How do these things sort of compute?
So epigenetics are the most used because it's the first technology that was developed to build clocks, to build these biological aging clocks. The problem with epigenetics is that it's really hard to change. We don't really know how to intervene in your epigenome. And the results, test-retest precision is oftentimes not so great. You could send your sample to different labs to do epigenetics. You may get 10 or 20 years of spread
in either direction.
We've seen some, and we prefer the youngest age that company gives us. So the test-retest precision is not super great. That's one, and two, it's not very actionable. And so that's the other piece that I don't feel very okay, it's great. I look younger or not so young, but what do I do with that? If I don't, I don't know how to change my epigenome. Whereas if I'm looking at labs, we have 120 years of research to move the needle and to bring your labs back to an optimal state. If you're looking at proteins for your inflammatory age clock, we know exactly how to modify those proteins. And there's a handful of them. So, there's pros and cons. When we look at the ability to predict chronological age, epigenetics are incredible. They're really good. But who wants to predict chronological age? Might as well ask you for your ID.
We already know it, right? You already know your ID. So this is fascinating. And then, we, of course, are we are positioned right now to offer this immune system age at the Holistic Heart Centers. And I think we're going to expand into really trying to understand the mortality risk and the organ age, because, there's such a huge connection between the aging process and the cardiovascular system. Cardiovascular aging essentially is aging, and a lot of other things follow. Now, you found something interesting. I remember your talk, when I approached you, you looked at astronauts, right? The people who've gone into space, and you found something interesting in that group. Can you comment about that?
Yeah, so NASA reached out to us about 5 years ago to start a collaboration because they've seen accelerated aging in astronauts. The astronauts that go in space maybe for a year or even a few weeks, they have signs, aspects of accelerated aging. And that really prompted my whole lab to move into a specific direction of space medicine. I care about astronauts, right? But
Sure.
I care more about the 8 billion people we have here on our planet, on Earth, that will never go to space. And so how do we use these learnings to improve healthy aging on the ground, on Earth?
Mm-hmm.
We took cell culture system that was developed by NASA that can put your cells in simulated 0G. And we can have a window for how you will be aging, because that causes aging in your cells.
In the cells, right.
Not in you, right? You take your cells. And so we have almost like a digital twin that you can accelerate the aging and learn from that, and then see yourself in the future. So we have biomarkers of future aging. These are all the hallmarks of aging we can actually predict based on microgravity. The paper was actually preprint today.
Wow, congrats.
Yeah, I'm going to send it to you, but it's in the review in a major journal. So you're going to see that we can predict with the microgravity how that person will age in each hallmark of aging. And that gives you a comparison with you against yourself. Not against the population, not against the population.
So the N of one, and some of my colleagues, because we have our early digital twin at the Holistic Heart Centers. We have something called Heartwell.ai, which is a simulation that allows us to understand the impact of the imaging, genetic, and biomarker and clinical parameters on cardiac risk down the line, right? It's only a simulation. We don't have enough information yet to do the full digital twin, but it is the beginning. And I always tell my colleagues is that the trials of the future are probably not going to be those huge randomized double-blind placebo-controlled trials. They're going to be a pragmatic N of one investigations that we will be able to do at scale, because, we will be able to do exactly what you're doing in time. So do you foresee this as another opportunity? Let's say, you'll get a swab of our cells and you'll put it in that interesting simulated environment, and it comes back and it'll say, hey, Regina, this is what's going to happen. We think you're going to die from X, Y, and Z, and then you better take care of it now.
So it's available. It's a product that's called Beyond Age. We have it with many different clinics and concierge medicine, and I'm hoping that you will incorporate it into your practice, because it's really a game changer to establish those risk factors that compare you against yourself in the future. So it's a very unique value
Very
proposition.
unique product. Amazing. So, we absolutely are incorporating this because we're standing up sort of like the whole longevity suite, and as you mentioned, it's sort of that second or third layer that gives our patients, honestly, I feel that it's the power. There are individuals who say, I may not wish to know about, some genetic markers. And even with something like this, that digital twin simulation of the impact, the mortality impact, and maybe even a timeline, that could be upsetting. But I certainly hope that most individuals will see it through the lens of empowerment, right? This is something that we can do. This is some preventive care. This is something that we can change.
The one thing I always like to say is biological aging is malleable. You can change the future of your health. You can control it. You can empower your subjects, your patients, and to do better and have a healthier lives.
100%, and be there for your family and friends. Well, Dr. David, this was immense. Learned a ton. I now have to run off and order my Beyond Age testing kit because now I need to know. And so, and we will be back with more of this once my test results actually land. Thank you. Thank you for tuning in to Own Your Heart Health with Dr. Regina Druz. This podcast is powered by Holistic Heart Centers. If you enjoyed the show, please rate and review us on your favorite podcast platform. To learn more about our services, visit holisticheartcenters.com and subscribe to our YouTube channel. The link is in the show notes. See you next week.